brief introduction about, good morning, Dr. Krishnakumar. A brief
Morning.
introduction. Uh, Dr. Krishnakumar is a senior principal scientist in the Environmental Technology Division at the National Institute for Interdisciplinary Science and Technology, Trivandrum. He leads the environment cleanup technology activities at his institute. He is also a professor in CSIR Academy. He developed a passion for the subject while pursuing postdoctoral studies at the National Institute for Environmental Studies in Japan under the Staff Fellowship from Government of Japan. His research focuses on bioprocesses for waste treatment and resource recovery, bioremediation, environmental impact of emerging contaminants, and microbial ecology. He has about 28 research publications besides two US and two European patents, and his gene bank submissions stand at 25 at HCPL. I welcome Dr. Krishnakumar to deliver his talk on field demonstration of a hybrid biophysical process for generating potable water from perchlorate contaminated abandoned community well. Over to you, Dr.
Thank you, ma'am. Greetings from CSIR Trivandrum, all those who are listening me. I'll share my slide first. First of all, let me thank the organizers for inviting me for this today international webinar. And the topic I'm going to discuss today is a field demonstration of a hybrid biophysical process for generating potable water from perchlorate contaminated abandoned community well. In fact, this is one of the activities CSN is pursuing maybe for the last 10 years, in which we were trying to find a sustainable solution for a pressing environmental and public health problems. The whole story started in 2012. During that period, as part of an ongoing project, we were screening drinking water samples from different states and union territories in India. We were trying to screen out emerging contaminants, particularly endocrine disrupting micropollutants. So in that study, what we came to know is that there is an emerging micropollutant which is called perchlorate. I'll come to later what is perchlorate. We can see the different states and the level of perchlorate. Very interestingly, states like Kerala and Tamil Nadu, we come across exceptionally high levels of perchlorate. For example, in Ernakulam districts, particularly at Aluva, the concentration was something like above 40,000 microgram per liter, where the international WHO guideline was only 70 microgram per liter. Similarly, in Trivandrum also at one place, we got more than 700, and in Tamil Nadu, particularly at Shivagashi, the concentration was around 4,600 microgram per liter. Since we got exceptionally high levels of this particular contaminant in well water samples, we did a number of samples. You can see the number of samples we have taken from different states at comparatively less, because we want to make sure that what we are observing is absolutely correct. So the exceptionally high levels of perchlorate against the WHO guideline. In fact, WHO guideline still it is 70 microgram per liter. In fact, it was the first report of massive contamination in India. And one specific observation was we found these high levels of perchlorate around places where it is handled in bulk, particularly like space R&D units and so on. So those who are not familiar with what is perchlorate ion, perchlorate is a toxic oxyanion, something similar to nitrate. There's one chlorine atom and 4 oxygen attached to it is an anion, toxic anion. It's a strong oxidizing agent. Because of its strong oxidizing properties, it's mainly used in arms and ammunition sector, as well as in space R&D units as an fuel oxidizing agent, where the fuels may be many times it is magnesium or alumni kind of metals, and these kind of chemicals were used as oxidizer for the fuel. Apart from these, many industries, including cracker manufacturing and even small-scale quarter units, those are making matchbox, are using perchlorate salts, either sodium, potassium, or magnesium salts of perchlorate. It's highly persistent, and its half-life is something like 100 years. And its natural degradation is very difficult. Why? Because I'll be coming in the coming slides. So the underlying factor, it's mainly used in AMS and emission sector, space R&D units, and its natural degradation is very difficult. And if you look at the toxicity of this particular ion, it mainly affects the thyroid gland functioning, leading to hypothyroidism. So what it does is it's competitively bind with the membrane proteins that normally helps in the transportation of iodine molecules into the thyroid gland follicle cells, where all the thyroid hormones T3, T4 also are synthesized. But when perchlorate ions are there in your blood, it competitively binds the membrane protein so that iodine transportation doesn't happen. So even if you take a lot of iodine through your food, the synthesis of thyroid gland hormones will not happen. So pregnant ladies, fetus, infants are the most vulnerable groups to this particular chemical toxicity, and it can even penetrate the placenta barrier. So hyperthyroidism has adverse implications for structural and functional brain development in fetus, infant, and child. A lot of animal studies have indicated that it can lead to arrested growth and arrested metamorphosis in amphibians and many animal models, even sex reversal, and in some studies, even tumor development has also been reported for this particular chemical. And if you look at the international guidelines, as I said, the WHO guideline for this particular chemical is only 70 microgram per liter in drinking water. And in many US states, they have brought down the permissible levels from 2 to 20 microgram per liter. And as per US EPA guideline, it was 24 ppb until 2019. And there are no guidelines in India so far for this particular toxic chemical. I said in Kerala, we observed exceptionally high level of perchlorate and one place in Ernakulam. This is near to Alua. This is a place, you are seeing the Google map of that particular area. So this place comes under the Kirmad Panchayat in Ernakulam districts. And in that Panchayat, there is one particular area called the Pulakkad Colony area, where extremely low-income family people are staying here. You can see large number of houses. And each building, sometimes two families are occupying because they are very low-income families. And all these people were depending on some community wells for their daily water requirement. And these are the three community wells. I labeled it as public well 1, public well 2, and public well 3. You can see the houses, densely packed houses. Nearly 200 families were there in that area. And these community wells are heavily contaminated. In 2012, when we initially did the analysis, the level was something like around 40,000 in PW1, and this was around 25,000 and so on. I'll be sharing those details in coming slides. So this is the place where we did the study. And since we have observed exceptionally high level of this particular chemical that can lead to thyroid dysfunction, we intimated the details to the public health department. They did a medical camp there. And this is some snapshot of the medical camp. And they found that an elevated or higher incidence of hypothyroidism among people exposed to contaminated well water. So they did a couple of medical camps, and the average hypothyroidism in Kerala population is something around 10%, and people were taking perchlorate contaminated water for quite some time. And since we brought out this, they stopped. In fact, they closed all those wells, and people were provided with alternate drinking water. And immediately after that, they did a medical camp. And the first medical camp, they found the thyroid TSH level is almost 18% higher than 18% in the public there. And but when they started drinking normal water, and there was another medical camp almost 2 months later, interestingly, we found that the TSH level from 80%, it came down to 10%, and that persisted. It clearly indicates that perchlorate is the culprit. And what I'm sharing here is the data points of TSH value. The green line indicates the normal TSH level, slightly above 4. And we found all the red data points indicates the values for camp number 1, and the white data points represent the TSH of people during camp number 2. So a large number of people were recovered after they have started taking perchlorate-free drinking water. So with this, we flagged that perchlorate is an emerging contaminant of high public health concern in India. So these are what you see here are the closed public wells. This is the PW1, PW2, and 3. And since 2014, all these wells are closed. People are not using these wells for the last 8 years. We were testing large number of well water samples in that area, and we were sharing those details to the panchayat so that the panchayat was providing alternate drinking water for the people in that area. So even after 10 years, the perchlorate level are high in most of the wells in that area. The contaminated fume getting diluted, spreading more area, and we have observed contamination up to 2-kilometer distance. So we were screening a large number of well water samples, and we as I mentioned, up to 2-kilometer distance, we could observe perchlorate level in well water samples. But the level was getting diluted so that some of the wells, the level was very low. So this table shows the perchlorate level from 2012 to 2021. As I said, initially it was 14,000, and in 2021 it was around 3,800. So this doesn't mean that it's getting attenuated naturally. It's getting diluted, and it's getting spread to more areas in different concentration levels. Now, having said a background about the public health issue and related environmental problems, what are the remedial options? In fact, Americans were the first to detect the health issues associated with perchlorate, and they started working on it from 1965 onwards. If you look at the different remediation approaches for perchlorate contamination, so this is the data from US Groundwater Remediation Research Team. They have a couple of full-scale plants operating for decontaminating perchlorate from drinking water. So you can see most of the approaches are biological methods. The physical methods, or in situ physical or ex situ chemical, whatever it is, it comes around 30%. So nearly 60 to 70% of the methods are biological methods where they're using microbial systems for decontaminating the perchlorate. So even if where they're using physical methods, either ultrafiltration, nanofiltration, or RO, the rejects or washed water, that will be with high concentration of perchlorate. There again, they have to depend on microbial system to decontaminate it. So the challenges here is you have to isolate potential bacteria that is capable of degrading the toxic chemical. You need to have intensive basic studies to identify key parameters and optimizing the same for process development. To be more specific, you should know the ideal substrate for the bacteria, the substrate perchlorate ratio, redox conditions, physical parameters. If there are any competitive inhibitors, how to address those things, the cofactors for those enzymes involved in the biochemical pathway, and the biomass. All these are important parameters that we should address when we develop a process like this. So if you look at the science of microbial perchlorate degradation, or in fact, it's a reduction reaction. I've just shown here as a cartoon form. This is a perchlorate reducing bacteria. So if you provide a electron-rich suitable substrate for the bacteria, the perchlorate will function as a terminal electron acceptor. It's simple anoxic respiration, just like what is happening in nitrate reduction or sulfate reduction. So the perchlorate will function as the terminal electron acceptor, itself getting reduced into chloride and oxygen. As we know, both chloride and oxygen are totally non-toxic. So this is a basic, very basic for microbial perchlorate reduction. In fact, this whole biochemical process is catalyzed by two enzymes. One is the perchlorate reductase, and the other one is chlorate dismutase. These both are membrane-bound enzymes. What it does is the perchlorate initially get reduced into chloride. One oxygen is removed, and subsequently it reduced to chloride, where one more oxygen is removed. And from chloride, 2 oxygen and 1 chlorine are reduced. As you please note here, this reaction, you're releasing oxygen. Oxygen evolving reactions are very rare in biological systems. So 2 enzymes, perchlorate reductase, which catalyzes the first step, and the last enzyme, chloride dismutase, catalyzes the last steps. And these are the corresponding genes according for these 2 enzymes. And if you talk about the substrate, that could be heterotrophic substrate like acetate, methanol, glucose, like that, or even autotrophic substrate like hydrogen, iron, or sulfur. are used by different perchlorate reducing bacteria. These perchlorated bacteria, generally we call them as PRBs. And in fact, these organisms are mostly ubiquitous. You can find them in any environment. And most of them, these organisms belong to beta-permeable bacteria. Even though these bacteria, as I said, are present in nature, why natural degradation doesn't happen? Because of these reasons. You need to have presence of these PRBs in sufficient number. Not only that, the availability of substrate is very important. The appropriate redox potential, that's very, very important. And the presence of competitive electron acceptors. If instead of perchlorate, competitive electron acceptors like simple oxygen or nitrate or sulfate is available, bacteria may use those them. In that condition also, this reaction will not happen. And because of these reasons, the natural degradation of perchlorate is not happening. That is why its half-life is something like 100 years in natural environment. So this is how we begin the process development, the perchlorate remediation bioprocess development stages. We start with environmental as well as bioreactor samples for isolating the bacteria. We start in small bottle experiment for enriching the microbial communities capable of reducing perchlorate. Then we went on scale up in Chemostat system, so something like fermenter, where we optimize all the parameters for perchlorate reduction. Parallelly, we were isolating bacteria. We were using, we were doing metabolic gene probing for identifying the bacteria, where we were mostly focusing on the two genes, PCRA and CLD genes, for identifying bacteria which are capable of reducing perchlorate. We were also using CCNS sequencing for identifying the bacteria. And all the potential cultures and the sequences were submitted to NCBA as well as IMTech culture collection for patenting purpose. So once we got all the parameters, then the major challenge was for translating these lab scale findings into a technology for field implementations. Initially, we went for pilot scale studies, and from those data, we scaled up the process for field demonstration. So the major parameters, as I said, suitable substrate is very important. What is the ratio of perchlorate to substrate for these reactions to happen? What is the ideal optimum redox potential for this process to happen? And again, the effect of competitive electron acceptors like oxygen, chlorate, or nitrate, physical parameters like salt, pH. And if the presence of heterotrophs, opportunity heterotrophs can occur in your system, in that case, how do you manage, how do you sustain the presence of your bacteria in the system? All those points were there. So these are a couple of our potential bacteria that expresses both the enzymes, so that these are the candidate organisms that we can adopt or depend for the perchlorate remediation system. Among these, the three bacteria are our magic bullets. We use in most of our treatment systems, we use these bacteria. One is Serratia marcescens, another is Halomonas species, and another is Bacillus cephrensis. So this is a semi-image of some of the microbes: Acinetobacter, Dichloromonas, Chlorospirillum, spirulline bacteria, Bacillus, Pseudomonas, and Serratia. I'll just elaborate the isolation of one of those bacteria. So this is a Chemostat, one of our very early studies during the development of the microbial system and optimizing the parameters. This is a Chemostat system. We are operating in fed batch mode. As you can see here on the wall of the Chemostat, there is a reddish biofilm flow. And what you see on the right side is a phase contrast image of the same biofilm. You can see a red patch. If you closely observe them, those you can see red spot. These are all microcolonies of Serratia marcescens, microcolonies. And when you isolate them, they form very clear, distinct colonies producing, which is very typical for some of these Serratia, pigment-producing isolate. This is an image of the same bacteria. In fact, this is the first report of perchlorate-reducing Serratia. And not only that, it's a first report of perchlorate-reducing gamma-protein bacteria, because most of the perchlorate-reducing bacteria reported previously belonged to beta-protein bacteria. This belongs to gamma-protein bacteria. And then Serratia is known as an opportunistic pathogen. And we were examining whether this candidate organism would have some risk. We did all the virulence tests, and we found it is safe to use in remediation systems. And the culture we deposited in MTCC, which with the specific number, and you can see the 16S DNA sequence number as well as the gene sequences for both the PCRA as well as the CLD gene sequence. Luckily, this bacteria was also expressing nitrate reduction, because even if you have nitrate in your contaminated water, that is not going to affect your perchlorate reduction. That's an added advantage when we use this bacteria. So this is a whole simplified process flow sheet. You have the bioreactor in which you augment it with your microbial system, which is a constituted culture of all your identified bacteria, mostly the 3 bacteria that I mentioned earlier. So you have the contaminated water, there will be the contaminated well water, you'll be pumping into the reactor, where the microorganisms reduce most of your perchlorate in the water. And whatever water which is coming out, make it a potable quality, you need to have a physical filtration system. Not only that, your target is bringing down the initial perchlorate concentration from few thousands to less than 70 microgram per liter. And the bioreactor treated water reaching to that level practically was not possible. For that purpose also, we have introduced a couple of filtration units. The purpose of microfiltration units was to filter out even some suspended solids or microbial cells that we can expect from any bioreactors. And not only that, we have an arrow module at the end for removing all the residual perchlorate in your sample. To extend the life of our modules, the MF unit or the microfiltration unit was helping. So we have a bioreactor first, followed by a microfiltration unit, and finally you have an RO unit. So whatever the reject and wash water that contains perchlorate from the MF unit or RO unit, you'll be pumping back to the bioreactor, making it a totally zero discharge process. And the water which is coming out from the RO unit was of potable quality. So this was the pilot scale treatment system. In fact, this study we were doing in 2018 period. This unit was capable of producing around 200 liter per day. You can see here, this is a simulated well water spiked with the perchlorate. In fact, we were trying with the 15,000 microgram per liter in the water, because in 2018, some of the wells at the site, at the actual site, the level of perchlorate was something like 15,000. So we decided to go for that concentration. The well water spiked with sodium or potassium salt of perchlorate at this particular level. And this is a bioreactor. This is a 100-liter bioreactor, fixed film bioreactor, anoxic, augmented with our specific proprietary microbial system. And the water which is coming out from the bioreactor, we're pumping into this an MF module, microfiltration module, to filter out all the solids. And so into an MF unit, from there into a couple of, in fact, 2 RO cartridges to make sure the potable quality of the final water coming out. So when we're doing these studies, we come across problems like membrane fouling, because whether it is microfiltration or RO membranes, you can expect membrane fouling when you continuously operate it sometimes. And we were trying to address those issues, and we found that backwashing as well as forward flushing, these are very simple approaches. We can manage the membrane fouling to a great extent so that the life of membrane can be extended and output water quality is not being compromised. And another problem was maintaining the redox potential, because for this biochemical reaction to happen, the redox potential should be something like -200 or less millivolt. And we have some methods to maintain at that level so that the perchlorate reduction happens in the system. And another problem what we observe are the substrate limitation, because you cannot limit or you cannot control other heterotrophs growing into the system, because we were providing sodium acetate as an electron-rich carbon source for the bacteria to grow. And whenever acetate is there, you can expect other heterotroph bacteria can also grow, and they will compete for the carbon source. And then in that case, we have to provide the stoichiometric requirement of acetate for perchlorate reduction, we know, but we have to provide slightly higher levels of perchlorate. All those optimization studies we done during this period. But after 2018, in fact, after that was COVID period, was not only that we were desperately looking for proper funding for taking this technology for field implementation. And we were successful in 2021 when we got a funding from the Jal Jeevan Mission. And that funding was for field demonstration of the technology. What you see here is the whole treatment system that we took to the field. We were operating this system in lab to avoid any delay in operation at the field. We were simulating all the field conditions, and we were operated this system in the lab for nearly 2 months. So what you see here, sorry. So you can see here, I'll show you in the next slide. Okay, so what we're doing is you can see a pipeline here coming. We have a collection tank here, 5,000 liter collection tank spiked with the perchlorate, around 3,000 microgram per liter. Because from 2018, when it reached to 2020, the actual level of perchlorate in some of the wells was around 3,000 and 1,000 range. So when we're doing these kind of studies, the concentration that we opted was only 3,000 microgram per liter. So we spiked actual well water with this much level of perchlorate, and we pump into an overhead tank. And you can see a small bottle here, which is a concentrated substrate for the bacteria. And we mix all together, and we were controlling the feed or the perchlorate contaminated water in a couple of bioreactors. So there are 2 bioreactors in parallel. Each reactor is 1,000 liter capacity. And from both the reactors, the treated water collected in a common tank. From there, it is pumped into the physical filtration system. So the major challenges here was scale up of the bacteria culture. So far, we are doing in small flask or small fermenter kind of thing. From that, you have to scale up to 2,000 liter bioreactor requirement. So that was a huge task, but successfully we could do that. And another tasks were maintaining the optimal conditions like the redox potential, maintaining the substrate perchlorate ratio, providing nutrients. All these challenges were there. So as I said, the inlet perchlorate concentration was around 3,000 microgram per liter. The substrate perchlorate ratio, we were maintaining at 1:5. The flow rate was 80 liter per hour, and the retention time, hydraulic retention time in the reactor was around 12.5 hours. So the reaction was so fast. We need only 12.5 hours HRT inside the bioreactor. So the both reactors were producing around 3,840 liters per day, and the bioreactor out water from 3,000, it came down to 100 microgram per liter. And the removal efficiency was nearly 99%. So nearly 99% of perchlorate was getting reduced in the reactors. So from the reactor out common tank, we were pumping into the ultrafiltration units and finally into RO module. This is a combined MF RO module here. It's a custom designed MF RO module based on our technical details.
Krishnakumar, just sorry
Yes.
for interfering. Just wanted to inform that we are reaching the time limit.
I'm finishing now. I'm finishing. I'll just skip all those things. And so this is the actual well where we implemented the process. This is an abandoned well. It was closed for the last 10 years. So the actual perchlorate level in this well was around 1,300 microgram, and the pH was acidic, maybe due to some organic infiltration. So the entire well water was pumped out, and the well was replenished, and in 12 hours, the same perchlorate level water was found in the well, indicating that they said entire groundwater plume was heavily contaminated. So this is the actual treatment unit, what we installed in a very limited space. The entire treatment system, you can see the bioreactors on the back side. This is a physical filtration system. So the installation we did in December last year, it's almost 1 year now. It's a fully automatic system. And this is a well from where we pump the water here, and the treated water we are distributing at this point. Okay, the trial unit we've operated in a trial basis for 3 weeks before we started distributing the water to the public. In January 14th last year, there was a small inauguration function. You can see the Panchayat officials and officials from the health department. And we were generating around 2,000 liter potable water per day, and the production cost was only 20 paise per liter. So from 2012 to 2016, these are the news about the contamination in that area. From that point in 2022, we could come up with a sustainable technology providing drinking water from an abandoned community well. So these are the publications from our side, starting from the surveillance, technology development, and all those things. And we also have a US patent for the process covering our patented, proprietary microbial system. This patent was granted in last July. So if you look at the major impact, we flagged perchlorate as an emerging contaminant in India. We also brought out the cause of a public health problem. Our intervention helped recover nearly 90% of thyroid affected people, and a large number of people were protected from health problem. We could handhold the panchayat and health department to address the issue. We sensitized bud handling units to take precautions to avoid environment release of perchlorate. And above all, we developed and implemented at field a sustainable solution for the problem. My acknowledgements mostly to the funding agency from the Department of Biotechnology, Kerala State Biotech Commission, because the analytical facility for doing perchlorate we could establish with the help of funding from the Kerala State Biotech Commission. That was in 2010, that period. We have a project from CSIR under the 12th five-year plan. And the recent funding is from Jal Jeevan Mission, the Ministry of Jal Shakti. All the directors during the course of this project, heads and colleagues of my division, all the research students and project staff in particular. And of course, the Kirmai Panchayat, the Primary Health Center, Indian Institute of Diabetics, Trivandrum for doing all the TSH analysis, and of course, the people at Kolkata. So the photo what you see here is the photo I have taken in 2014. And these are my initial students who worked in the project. And what you see on the background is a pond. It's a community pond, which is also heavily contaminated with the perchlorate, and still it is contaminated. And it's a major challenge we don't know how to address, because we still have continuous infiltration of perchlorate contaminated water into that pond. That I'll end it here. Thank you for your listening. I'll be happy to answer any questions. Thank you.
Thank you, Dr. Krishnakumar, for taking up a very important, socially relevant problem of perchlorate contamination in our community. And not only that, you have sensitized the health authorities, notified the people, and also developed measures, so that you can remove the perchlorate from many of their wells, and then implemented that in a small scale, and scale up to a larger scale, and then finally installed that in the field and have shown that and supported the people and the community to reduce the perchlorate contamination. Thank you very much. That's a very interesting work that you have shared with us. Just curious that you said finally at the end of your talk, you said still there is perchlorate contamination into the wells. So have we identified what was the real cause of the contamination? Because you said it's confined to Alua region. But is there any, what actually is leading to that contamination?
Yeah. In fact, there is a bulk perchlorate producing industry at that place. Yeah, that is the main source.
So should we not, sensitize those people?
We have sensitized them also. We have sensitized them also. They are taking all the precautions, because even they may not be doing it purposefully. It could be an accident spill or something like that. But they know the seriousness of the problem.
Okay.
So congratulations for taking it to the field, to the people. So that makes a difference in the research. I mean, when you get a lot of satisfaction,
Definitely
got that, and to take it to the people. And I just wanted to suggest something that relates to you. In Kollam, the KMML, the titanium factory, the wells around that place is heavily contaminated with iron oxide. Probably we
know that, sir. Sir, we know that. We know the problem.
Yeah, probably that will be a nice effort to that. Yeah, thank you. Dr. Nitu, it's up to you. Shall we go ahead with the next?
Excuse me, sir.
Hello? Yeah.
Yes.
Hello. There are questions in the
Yes.
chat box. If you can.
Yeah. Could you please read it if you don't mind?
Dr. Nitu, can you just read from the chat box? There are some questions over there.
Just one second.
Should I read? Okay.
Yeah. Please go ahead. Please read it if you
The question was, is this applicable in large scales because of cost and facilities, as we here speak about wells and large quantities of contaminated water? So I think cost-effective implementation is what he's looking for.
Yeah, I have specified the cost of production of water was only 20 paise per liter. because this is purely an anopsic system, and you don't need too much of energy input. And you need only a small pump for pumping the water from the well to the outside. The rest is even the 20 paisa, what I said, that including the operator charge. So if you go for a large scale, I mean, this cost will still come down.
Okay, thank you, Dr. Krishnakumar, once again.
Thank you. Yeah. Thank you all.
Thank you. And
Yeah,
the
just
next
wanted to make sure.
yeah, please, Dr. Nidhi. Yeah.
And I just wanted to make sure that Dr. Sonia is online. So.
Yeah.